Welcome to another edition of Ivory Tower
Grumpy Science, where you can tell that this is a completely different week. We do this.
Oh yeah,
we thoroughly research a week apart. It's just that
I've got two shirts just like this.
Actually, it's been a week, so I had to wash the same shirt and I wash my hat at least once a year. But
only when it has to be done. Yeah.
Yep. So anyway, we've got our multimedia center here pulled up, and last week we talked about lateral inhibition, if you remember. And you know, the thing we want to talk about a little bit here is that what happens when things go wrong. And we gave the example of how if we're looking at sine wave by sine wave, pulse by pulse, what happens to a whole class of receptors, NMDA receptors, when they quit working. Well, first of all, when they're working right, they start out being bored, and then because they're voltage dependent, as the excitation goes up,
you build up.
They build up
a response. It's like somebody poking you to wake you up. You just get
right, and plasticity takes a big advantage of this because it starts building up these patterns of responses to different stimuli, and it builds that up both by turning on NMDA receptors and NMDA receptors getting turned off.
So is it correct to say, because I don't know, Mark's the brain scientist, right, that the basic process of learning requires this kind of dynamic?
Absolutely.
So I think it's as fundamental as learning something, anything, a skill or a fact. You have to have at least this level of neuroplasticity. Right?
Absolutely.
And is it also correct to say that, like, if you don't have that, it can lead to things like autism and things like that? You don't have the ability to.
Interesting that you asked, Bob.
I just
but while you're asking,
question,
we can segue off into another. This reminded me of something, and we never get off topic.
Oh, no, man, straight as an arrow. These things are practiced, right?
Yeah.
That's why we never have to.
So you're absolutely right when we talk about the different mechanisms we're going to talk about and how we measure them. And by the way, this measure is with your simultaneous amplitude discrimination measure, and it shows up in your accuracy metric on the brain gauge. Because what we're doing is we're looking at how these inputs interact, and basically the idea is all of cortex acting like this. And yes, learning is very tied to this particular beast of a mechanism, and it plays a really important role in it. If you don't adapt, you're not learning very well.
Yeah.
Well, we get questions all the time from people. Oh, well, do you do learning or memory tests? I was like, no. What we're doing is we're looking at the basic fundamental
mechanisms of learning and memory. Right?
Exactly.
Not sort of like, do you remember the number of stars in this box or something?
And there've been a number of articles lately on these cognitive tests. The cognitive tests are used for concussion, and apparently, shock of shocks.
No, he's not cynical.
No, some of these college athletes who are taking these cognitive online tests for concussion testing are learning the proper way to answer all these tests and are scoring extremely well. And so right after they have a concussion, they score perfectly well on these tests.
They've learned how to fool the test, right?
So anyway,
it's because they're not testing anything fundamental. They're testing something that's more like at a higher level learnable. Right?
Yeah, and that was kind of why I stayed out of psychology.
Well,
but, when I was an undergrad,
not basic enough. Yeah.
I wanted to understand basic mechanisms. So anyway, last week we talked about lateral inhibition. This week we're going to talk about feed-forward inhibition. And these are, by the way, both requests on demand. Someone asked if we would.
Yeah, we don't just talk about this because it's fun and cool and nerdy. We actually are responding to people's questions.
Yeah, which is amazing because we're always shocked that anybody.
How many times in your life has a scientist actually posted something in response to a question? Probably not too many.
Anyway, so do you know what feed-forward inhibition is? Say we were talking about equivalent circuits and stuff.
Yeah.
So what is feed-forward inhibition in an equivalent circuit or in an electrical circuit?
Well, it's when you're not using feedback to control the input. You're sending a signal forward to suppress the output.
Right. So Here you're gonna have to draw again. Let's
Got
draw it on
boxes
black.
for you. Okay.
I need a box about that big,
All right.
and we need some space. So
You need one box or two?
I just one.
All
I'm
right.
just gonna do feed forward inhibition. Now we want
That's
an
all you're gonna
input.
get. All right. So got an input.
Yep. And an output.
There's your output.
See, now we got room for a graph on the output side.
Right. See, I did that specifically because I know where he's headed with this.
Okay.
I'm a
So
scientist.
this is a clump of cells, and we're just going to think in terms of what is a clump of cells going to do when you give it like a lot of excitation. What happens over here is, tell you what, let's
You draw it because I'm gonna mess it up for sure.
I'm going to mess it up because
I gave you some nice boxes. Now take it from there.
I'm making this up as I go along. So, that whole concept thing.
Making up facts.
Big feed forward inhibition.
Like alternative facts?
Yeah, that's
Who
right.
does that? Who does that?
I don't know. I can't imagine.
It does come from the top, though.
Yeah, so we can't
if you
get
don't
in
know
trouble.
the facts, make them up because that's just as legitimate, to our knowledge.
Okay,
base.
so let's say feed forward inhibition. What it does. Oh, this should have been red.
Yeah.
But it's gonna basically turn things off upstream. So you got this input here. Now we'll put a plus sign there. Everything's wrong colors. So let's look. We got all this activity here, and this guy's churning. All these cells in here are churning away, doing what they want, and just having a great time. And they think, and there's modules here, and there's modules here, there's modules all around them, sending them all kinds of inputs. They're getting all kinds of random inputs from other places, and from here and here and here.
Yeah.
Well, and so all this stuff's going on, and then the feed forward inhibition just says, I think I'll just shun it. I'm just going to bring that to zero. Who cares? Okay.
Right.
Well, there's a different there to get into the real particulars of how one type of feed forward inhibition works in the brain. You have to think about there's a class of cells called NGF cells. NGF, neurogliaform cells, and what these guys do is they provide inhibition to pyramidal cells. Now, pyramidal cells are sort of the mainstream. Oh, they got all this excitation going on here, and you get excitation from the thalamus or thalamocortical projections. But these NGF cells, when they receive excitation from the thalamus, they try to turn off pyramidal cells.
Yeah, and there's actually a good reason for that. Well, there's many good reasons for that, but the engineering reason for that is that if you have positive output, an output that builds on itself and kind of goes out of control, oftentimes you need to put that inside of an inhibitory loop. Otherwise, it'll just go out of control. So a lot of times you have to wrap up a positive feedback loop inside a negative.
Yeah. Well, in this case, they play a huge role when you deliver stimuli that you don't really want to care about.
Right.
Okay, so you get weak stimuli,
Dad,
and that can be
that
weak stimuli can be from you got
There you
kids
go.
in the back of the car.
Dad, are we there yet? Are we there yet?
After a while, you don't even hear it. It just goes into a different category.
There you go.
And actually, on a different scale, that's what's going on. But what happens is you get, let's say it's a really weak stimulus. And I drew it lightly in case you can't see it because it's a weak stimulus. You get a weak stimulus into this NGF cell, neurogliaform cell, which is inhibitory, turning off this. Well, it's strong enough, the stimulus is strong enough to turn off the neurogliaform cell, and it turns on the neurogliaform cell, which turns off the pyramidal cells, which are the ones distributing all this excitation. In other words, it's going up here and it just says, yeah, you're getting cut off. Basically, you've had enough.
So weak signal in here has a stronger effect downstream.
Exactly.
Which we model with electrically we model as like a transistor.
Exactly. Now, do you know what is so you kind of go, so what? Who cares?
I say so what all the time.
Yeah.
Irritates him. That's why I do it.
So the interesting part of this is that this is GABA B mediated. Okay, so we'll write this on the board. GABA.
He's already degenerating into illegible chicken scratch. That's
well,
okay. This is Mark.
You didn't want to write.
I wanted him to, carry the day here.
Anyway, so these are these provide GABA B mediated inhibition onto the pyramidal cells. Now, significance of that, normally like that lateral inhibition we talked about, that's usually GABA A mediated. So if the interactions that come with lateral inhibition, they're mostly GABA A mediated. GABA B mediation. So how do you detect that? Well, the only way to detect the only functional measure we know about for GABA B mediation is with the brain gauge. Otherwise, you can't do that with spectroscopy, and you can't you can do histology. So post-mortem you could do that.
A lot of things you can learn about the brain once you've decided to take it apart.
Yeah. So what happens if you deliver a tiny input over here, and what happens is basically this, let's say the input, let's say what the input looks like. Input's really weak, slowly builds up. Well, this input here, the output that based on this slow ramping stimulus, or something that's your kids are yelling louder and louder. When do we get there? All of a sudden the loudest, and then they start saying, "I gotta go to the bathroom." That's getting louder.
Stop touching me! Stop touching me! He's pinching me! He's pinching me! So
the stimulus is getting bigger and bigger, but the initial output is nothing. Stimulus keeps getting bigger. Well, what happens when this stimulus is getting bigger? Well, it's already suppressed the cell. So it's still ignoring it, but it still hasn't reached threshold yet. It still hasn't gotten to this. Oh, you should have drawn that one.
Yeah.
Okay.
A little bit of a disaster.
Anyway, when this stimulus finally hits threshold, then this output here, when the input finally gets high enough so that the pyramidal cells turn on and don't care what the NGF cells are yelling and screaming and turning things off, or the inputs are, in other words, kids are yelling, they're beating each other up, they've got to go to the bathroom, and you've got a huge migraine. You stop the car, and finally, the inputs to the pyramidal cells just overcome this. And so rather than getting a slow ramp of input, slow steady ramp, you get a step function. Boom. Yep. Just like that. Boom. All of a sudden gets wrong. Gets screwed up. Well, that's part of the story. Remember, we talked GABA A, lateral inhibition. GABA B, feed-forward inhibition. That doesn't mean that there's no lateral inhibition, no GABA A in the feed-forward inhibition, and no GABA B in lateral inhibition, but they're the main players in those. And how do you separate that out? Well, we deliver a really weak stimulus or condition. We do a test where we condition with a really weak stimulus, and it makes thresholds go way up. And then you basically get a step function. So this is one of the things in autism, behaviorally, it can be explained by this, is that some kids with autism, they basically ignore, and then all of a sudden, in other words, they ignore. They're very hyposensitive. They don't feel anything. Then all of a sudden, it shoots way up, and they're very hypersensitive. All right. Now, there's another thing about the NGF cells that's extremely important. I bet you know what this is. We talked about it before.
I'm not going to steal your thunder.
Okay.
Go right ahead.
Anyway, when we study basically feed-forward inhibition and kids with autism, we found that feed-forward inhibition is extremely low. So they don't have that gating mechanism for knocking out the superfluous inputs.
Right.
Okay.
They don't filter.
Another group also has low feed-forward inhibition, and that's people with chronic pain.
Mm-hmm.
Now, what would be really cool is if you could study a group that had really high levels of feed-forward inhibition. And it turns out in 2014, there was a paper published that definitively proved that neuroglialiform cells are the only cells in the brain that produce insulin. Now, a lot of diabetes researchers weren't even aware of that.
Right.
brain had been producing the insulin. But one thing we do
you
talk
don't think
about,
of the brain when you're thinking of insulin, right? You think of pancreas. Right?
yeah, and diabetes researchers basically don't talk to neuroscientists, but neuroscientists have been speculating on this, on insulin being produced in the brain since 1970s. That's how far back we found papers where they said
1980s or 90s.
You know, but if you go to most medical school textbooks, you see a nice little neat feedback loop with the pancreas. Pancreas produces insulin. Insulin goes in the bloodstream. Pancreas detects insulin and glucose and then generates.
Very likely that the brain is controlling that.
And somewhere in the hypothalamus, it looks like there are some things that, by the way, seem to be GABA mediated. Whether it's GABA A or GABA B, I don't know. But it looks like GABA plays a role in hypothalamic control of insulin production in the pancreas. But so what we did was we said, well, if feed-forward inhibition is really high, where would feed-forward inhibition be high, or what population would be high in? We guessed, well, if diabetics have anything change in their brain, it would be the insulin-producing cells, and that would have an impact on feed-forward inhibition. Mm-hmm. So we tested a whole bunch of diabetics, and this was not an NIH-sponsored study, so we could go ahead and do it on our own, and found out that diabetics had 3 times the feed-forward inhibition of healthy controls. And so completely polar opposite. In other words, you can have too much of something or you can have not enough.
Yeah, and a lot of times there are feedback loops that involve the brain for things that we don't think of as brain related.
Right.
In this case, a metabolic master control.
And the reason of that is that if you have a cell line that is producing insulin and it's in the brain of a subject that needs more insulin, well, it starts becoming hyperactive but it also had other functions. It also has that GABA B mediated feed-forward inhibition, which it's also getting hyperactive as a consequence. That would be called maladaptive plasticity, where
that's what I would call
the brain, and there are places in the brain that have 10 to 100 times more insulin than the bloodstream. Mm-hmm. So anyway, there's a lot of feedback loops, and since Bob made me start drawing, I'm not going to draw them. But there's a lot of feedback loops in the brain, and we'll say that for another talk.
Yeah, absolutely.
But basically, the things to remember are.
Did we say anything grumpy this time?
No.
Oh man,
I guess we could.
The grumpy to science ratio is
way
off kilter here. This is way too sciencey.
Yeah, way too sciencey. Sorry about that.
Really grumpy right now.
All right, so we can talk about maladaptive plasticity, and we'll bring this back in.
So maladaptive plasticity is when your brain makes a change that's not good for you, right?
Right, and that's what happened. That's one of the things, and we'll talk about that more later, about both chronic pain and how that's maladaptive plasticity or chronic inflammation, and the two go hand in hand. Yeah, but absolutely. Those things, basically, I guess we could come up with some everyday layman examples that are pretty grumpy about what happens when, it's sort of like when a tick starts growing, latches onto you. It's it's going to eventually die, but it ain't helping you. And that's a terrible example of maladaptive plasticity.
Well, I guess
or maybe an administrator might. Well, actually, maladaptive plasticity is an example of that. Is something like institutional review boards. They eventually, at the very beginning, they were.
When people fall into bad habits, when they fall into bad habits, like feeding back to each other, like married people or something.
Well, the maladaptive plasticity is where feedback loops aren't getting checked.
Right.
And administrators are really a good example of that. And so where we've had this growth of the 3-to-1 faculty to administrator ratio that morphed into 1 to 3. Where the administrators started out where faculty ask administrators to do things like purchase or check research protocols or check, help them with that.
Back in the day when there was typing pools, and you had to ask somebody to type something for you. That was a reality back 40 years ago.
Yeah.
So,
but basically, when administration gets too large, they don't have any controls.
Well, it's sort of like the Republican argument about central government. You know, it's government out of control. There's no other government sort of suppressing the growth of the first government. That's the problem. And bureaucracies tend to grow. They don't get smaller. People don't say, oh, we need less people in this department. That's okay.
But being apolitical, bureaucracies have consistently grown in the federal government, no matter who's in charge.
Well, that's the thing. So I'm nonpartisan about it. I mean, it's a maladaptive plasticity that we see in government, where they tend to adapt to things by forming new committees and hiring more bureaucrats.
Right? Well, and forming new rules people have to follow, and then they need more bureaucrats to enforce those rules.
Yeah. So,
yeah, it's kind of a...
maladaptive.
That's maladaptive.
There you go.
But your brain, unfortunately,
your
it does
brain
the same
can do
things.
that. It can rewire itself. And this is something that I was reading a paper about this recently. I don't recall the author right now, but it was like a 2017 review paper on stroke, because all of a sudden I'm interested in that for no particular reason. And a big part of what happens after a stroke is neuroplasticity.
Yep.
And a big problem with that is that not all neuroplastic responses to stroke are adaptive. Some of them are maladaptive.
Mm-hmm.
Some people, for example, develop chronic pain.
Yep.
So, anyway, I think that's probably good enough.
Yeah.
You know, you got by with very little grumpiness here.
Yeah, but
we'll think just enough to call it grumpy science instead of welcome to science.
All right.
And who would watch that anyway? I mean, when there's all these great grumpy cat videos that you could skip over to,
yep.
we have to ride in the coattails of something that people will watch. At least two of you. All right. Anything else?
That's it.
All right. See you later.
See ya.